Good afternoon and welcome to the closing session of the 2020 edition of the HiTech program. My name is Pedro Phyergy. I'm the General Manager of HiTech, and in the next few minutes, I'm going to walk you through what HiTech is and what is the program for this afternoon. So let me start by explaining what HiTech is. So we are a not-for-profit association that was founded in 2017 by a group of 19 private companies. And currently we are 24 members. Our mission is to foster the creation of social and economic value from research and development activities by bridging the gap between science and the market. And to do this, we are grounded on a collaborative network that leverages a platform to link researchers and business community. So these are our HiTech members, which are, as I said before, 24 companies from a diverse range of business sectors. Why we came together to do what we do? Well, in the last 2 decades, Portugal has seen a big acceleration of the development in science and technology. And as you can see from the blue line in the chart, it doubled the investment in science and technology, doubled over the last 20 years as a percentage of the GDP. And because of this increase in investment, the number of full-time equivalent researchers, which is your slide, basically trebled as a proportion of the population, and the number of papers in relevant international scientific journals increased 5 times. So the knowledge that is being created by the science and technology institutions has increased over the years. If we take into account the data from the last European Innovation Scoreboard, which is the statistical publication that has more updated information on the European Union countries, we can see that Portugal, in the 4 indicators that relate to the production of knowledge, compares reasonably well with the other 26 European countries. 100 being the average of all the countries. We are significantly better in international scientific publications and are very close to the average in all other indicators. However, if we take into account the indicators of the scoreboard that show the mobilization of that knowledge that's created by science and technology, if we take into account the mobilization of that knowledge, we compare much poorly to the other 27 European countries. There's one indicator in which we are better than the average, which is the employment in fast-growing enterprises. But in all other indicators, we perform comparatively less well. So if we all agree that for a country to become competitive, we need to move from an economy that's based on resource exploitation to one that's sustained on knowledge, and that to accomplish that, we need to mobilize the knowledge that is produced through research and development and use it to accelerate innovation in the business sector. So one needs to increase cooperation activities between the stakeholders of this knowledge production and mobilization system in such a way that businesses can access technological resources that are needed to develop innovations. However, the cooperation between the research and development organizations that produce knowledge and businesses that can mobilize that knowledge needs to overcome several difficulties or obstacles, namely cultural and institutional differences, different regulatory frameworks, and in some cases, geographic distance. So to overcome these challenges, it is necessary to build bridges between these different institutional logics that have a set of conflicting rules and conflicting norms. And that's where the role of organizations like HiTech that are called innovation intermediaries is key to break these barriers and build these bridges. So how do we do this? Well, first of all, let me try to make as clear as possible that HiTech is focused on innovation that arises from technologies grounded on research or science. So this is different from other types of innovation, namely innovation based on business models, which we do not do. So we are stubbornly focused on innovation from science. And to start building those bridges that are needed between science and businesses, we start by providing skills to researchers in knowledge valorization that enable them to link science and technology to product and market needs, and to better communicate science to a business audience, which usually it's one difficulty that scientists have because the language is totally different between these two different fields. So that's what we do in HiTech, and I will describe a little bit later how this is done when I start talking about this afternoon session. The second thing that's required is to enable innovation by connecting the producers and users of knowledge and bridging a gap between them. So that's done in another of the HiTech activities, namely HiTech Startup. And in HiTech's startup, what we do is to support a group of selected teams and projects from HiTech to either create science-based startups, looking for business partners in order to advance the technologies through co-collaboration or through collaboration projects, and also looking or helping teams looking for potential licensors of the technologies. So since we have been doing, the team has been doing this for the last 17 years now in HiTech, And before that, in a different setting, we have created a large network that now has more than 650 researchers whose skills we have mapped out and we can use to deploy their knowledge to the business community. So this network helps us facilitating the access of businesses to external knowledge and facilitate the absorption of that knowledge. This is done in another of HiTech initiatives called HiSolve, in which basically we discuss the technological challenges that our members companies have, and then we look to address these challenges by connecting these member companies with the researchers in HiTech network. So now let's talk a bit about HiTech, whose closing session you are attending today. So HiTech has two stages. The first stage is called HiTech 1, and the goal of this stage is to provide researchers skills in technology commercialization, to support the development and validation of a value proposition for a product concept that's grounded on the technology the teams bring to the program, and finally to screen breakthrough technologies in the fields of science and engineering. The program ran from June to July for 8 weeks. It's a hands-on experience for participants, an immersive experience in which the teams get training in business development and work with mentors to develop and validate the value proposition for the product concept. HiTech 1 will start again next year, as always, and pre-registrations are open for researchers that want to participate in the program in the HiTech website. The second stage of the program is called HiTech 2. And in this second stage, we provide an hands-on experience to the teams that participate to further validate the product concept and value proposition, and then develop a business case around the product concept. And it is the pitch of this business case that you are going to see later on when the teams start presenting. The teams are supervised by and helped by a group of business mentors and advisors that support in the project development, and we facilitate networking with companies in order to help validate the technology. So some of these teams will move forward for the HiTech startup program, in which, as I said before, I help the teams on a one-to-one basis, either by helping them creating a startup if they are entrepreneurial minded, or we help them find partners to start collaborative projects to move the technology forward, or in some cases, we may help them looking for potential licensors. So this is the team that it was behind HiTech 2, or still is behind HiTech 2 until we finish the session. I would like to thank all the team, and in particular, Roger Debo, which was indefatigable, helping us preparing the teams for this final session. Roger is currently very far away from us, so, but although it's 5 a.m. where he is watching this session. So Roger, thanks a lot for, once again, for the help. Of course, that we are also indebted to the mentors that supported the teams throughout HiTech 2 for their great work that they have done, and the work that was done just for the fun of it, because they help us on a pro bono basis. So we have 17 presenting, 25 researchers. So these are the teams that you will see later in the afternoon in different fields, scientific fields. All of them are researchers that we believe will help changing some of their research mindsets. So for this afternoon session, projects will start presenting immediately after I finish this talk. We'll have a break, a 10-minute break at 4:15. Then we'll continue with project presentations, and then we will have a small, more formal closing session in the end. So each presentation is 7 minutes, and we will have 5 minutes of Q&A. So everyone that's attending can participate in the Q&A by accessing the Q&A button that's in the bottom part of Zoom. I think for everyone is in the bottom part, and you can ask the questions by Q&A. Of course, I'd like to thank all the HiTech member companies for supporting the program, and in particular, the bank, BBVA, for a special support in this closing session. So now I will handle the session to Olímpio Torre, who is the team member that coordinated the HiTech 2 program, and he will be the host for the remainder of the afternoon. So I hope everyone enjoys the team presentation, and Olímpio, now it's up to you.
Thank you, Pedro. Welcome, everyone. Welcome to the final session of HiTech 2. This has been a long journey for our teams. We have a great set of teams, as you've seen from Pedro's presentation. And it's now time for us to start giving them the opportunity to show us what they've developed throughout these 9 weeks. The first team that I would like to call has been trying to help us save babies. That has been the key note on this team. And this team is called MetaboFind. And to introduce us to this team, I'd like to call Anna Gil to please present. So first, Anna would like to please start sharing your screen. And whenever you are ready, you can start your presentation. Now, so small lag here.
Just let me check it's everything okay. Sorry about this.
waiting.
What is happening? Sorry.
So while Anna tries to solve the technical issues of connecting, I'll get the chance to introduce Daniela and Tatiana. They are the two other team members who have worked hard at making this project a reality. And also, I'll tell you a little bit about the project. So I'll tease a little bit. So there is one key issue that affects a lot of ladies who are pregnant. And this team has found a way to help them to find that and to enable doctors to aid them. So now that we have Anna with us, let's see the presentation. Anna, please share your screen and start whenever you want.
All right, thank you. Okay, thank you, Alipiu, again. So my name is Anna Gil, and I'm speaking on behalf of the MetaboFind team. Our motto is our health in our metabolic fingerprint. And the service we propose today is named Natality Plus. It is all about preterm birth, which is birth occurring before 37 weeks. It affects 10% of pregnancies and it causes 6% of mortality in the whole world. It is the main cause of mortality in very young children, and it also causes morbidity later in life. In fact, these later aspects can lead to very serious consequences, including cerebral palsy. The global economic load of PTB is 47,000 euros per PTB newborn, and it amounts to 22 billion euros of total costs per year in the US alone. We've talked with several doctors and they all concurred that PTB diagnosis is the main clinical challenge today in prenatal care. There are some methods in place, including physical examination, which is, however, subjective and non-consensual. Other methods, including ultrasound, are still very high in false positives. Recent methods, for instance, measuring fetal protein called fibronectin, are still very low in true positives, and they seem to be effective only a few days before PTB. In any case, these methods are only applied to high-risk symptomatic women who amount to a third of the whole. If they were to be applied to everyone, doctors have told us that the benefit to cost ratio would be too small. So Natality Plus comes in based on our discovery that metabolic alterations measured in the urine of pregnant women can predict PTB with at least 10 weeks in advance and with at least 80% specificity and sensitivity. So the concept is to offer a personalized service for early screening of PTB markers in the urine of all pregnant women. The name of our technology is NMR metabolomics, and our workflow starts on the left here with urine collection in the routine medical checks. Samples are sent for NMR analysis, producing a very complex record which expresses hundreds of metabolites in the sample. This is fed into statistical analysis, and this will produce a database and help us detect PTB markers. These are sets of compounds which are specific for different regions and different countries. And all this workflow will culminate into a PTB risk report, which can look something like this, with an easy to read gauge of PTB risk level. This report will be sent to doctors who can then increase the frequency of medical checks, ultrasounds, microbial checks, hormones administration schemes, and prescribe early rest. In terms of market analysis, our segments are national health systems, private hospitals, and of course, doctors. If we consider the PTB worldwide costs, the big chunk of this pie is related to prenatal and early birth care, and this is where we will act. In fact, we will act on this particular market of PTB prenatal testing. This is a market on the rise, and it's forecast to reach just over 1 billion euros in 2025. We also want to note that if we take into account the number of births in Portugal, which is part of our entry market, and the associated costs, if we come in with Natality Plus costing 400 euros per unit, there would be an investment of 34 million, with additional follow-up adding 12 million. Even then, we can compute the savings, which would be very considerable, just under 350 million, and this is in Portugal alone. Our business model is a B2B model, level 2, starting with an order, proceeding with urine collection and shipment, initially to a single analysis site, which then should evolve to a multi-site format. Our service will offer analysis, statistics, database consultation, and a PTB risk report for 400 euros. As I mentioned before, this report will be sent back to the doctors. Our pipeline starts with marker validation in Portugal in year 1, scaling up to the rest of the Iberian Peninsula in year 2. We want to enter the market in year 3 in our peninsula and scale up to the market in EU and US from year 4. At the same time, we want to start commercializing markers for other prenatal diseases. From year 6, we want to start selling markers for long-term PTB complications. This is prognosis. And from year 8, we want to scale up to underdeveloped countries where this problem is even more serious. At the same time, we want to offer similar services in other diseases. In terms of IP strategy, we'll be protecting predictive PTB markers for different regions and countries, initially through trade secret, and later on there will be scope for patent filing. In terms of our financial analysis, we predict we'll meet our break-even point in year 2 after market entry, and that we will need these funds for the first 3 years to start up this business. We predict these values in terms of sales volumes, and during the first 5 years, during which we will increasingly be increasing the number of equipments dedicated to this, not only to services, but also dedicated to continuous validation. So there are 3 of us in the team. There's myself, I'm a professor of chemistry in Aveiro. Daniela Duarte and Tatiana Carneiro are both biochemists. They're doing their PhDs in metabolomics, in prenatal health and in cancer respectively. And we're very lucky to have these 3 medical advisors with us. And I want to finish with our belief that Natality Plus will be saving new lives around the world, going increasingly from the situations on the left to normal term babies on the right. Thank you very much for your attention today.
Thank you, Anna, for your presentation. Great. And right on time. So, good job. So now we'll start the Q&A session. Like Pedro announced in the beginning, it is going to be 5 minutes, and we already have some questions popping up, and we'll start by, well, chronological order. So the first question was posed by Antonio Diniz is, are you assuming that we will prevent all PTB in your calculation about the benefit from your diagnostic?
Maybe I can answer that, or maybe Daniela can answer that.
Okay, hello. It is a good question indeed. In fact, even if you only detected 50% of PTB cases early, we'll have a huge amount of savings in Portugal alone. That would be around 150 million euros.
Okay.
Yes.
So, anyone else add anything else?
No, so far we predict that 50% is the very minimum, and we do expect to be able to predict above 80% of the cases with the data we already have.
So another question here is from Giselle, regulatory pathway. So how do you see going from where you are into the market in terms of regulation?
Okay, this is one of the barriers to entry we have identified. I don't know if Tatiana wants to say something about this.
Sorry, I didn't hear the answer.
Regulation.
Okay. Yeah, it's one of our barriers to entry that we identify, although we think that will not be a problem since urine collection is completely non-invasive and we will have all the care with the data protection. So we are not worried about that.
But it will have to be considered.
Of course.
Okay, so Ana Gonçalves is asking a question regarding why you think our benefits don't make a patent from the beginning? Why aren't you aiming at having a patent right from the beginning?
Mm-hmm. We considering a couple of proxy examples in order to decide how to proceed, and we've seen that there are a couple of companies working on similar services, and they keep their developments under trade secrets, which I think it makes all sense because we are using a piece of equipment and a technology which many people have, but only us really at the moment know how to explore it for this end. So we want to keep it secret until we are safe and we can bit by bit reveal bits of our knowledge in the form of patents.
Okay, good. Now we have a question that comes from far, far, far away. So if you remember, Angus Kingham was one of the mentors that helped you in the HiTech one. So Angus is posing the following question. Very good presentation, and I always like the statistical analysis of multiple markets. But he doesn't think that you are clear enough regarding the action taken after the diagnosis. So what are the next steps after diagnosis to improve birth outcomes?
Okay, maybe I can answer this. We have talked about this with several doctors to know exactly what would change in the clinic if we actually gave them a report with some extra information. And so they were very clear that they would keep that lady under much more frequent surveillance. They could afford then doing some extra ultrasounds and microbial checks for infections, which is a usual source of PTB, origin of PTB events, and prescribe early rest straight away, and perhaps consider some hormone administration schemes. This is all personalized. It would depend completely on the person, but these are the tools that you'd have at hand and then could use. At least, if not to prevent, to delay the event, which would already be very important.
So we are now on the time limit, but we'll have time for at least one more question. What is the business model and who is the client? Is a question asked by João Milones.
Okay. Tatiana, would you like to answer that?
Our business model, of course, thank you for your question. It's a B2B service, and our customers will be the national system with a huge and important role of doctors being prescribers of our services.
I may add that private hospitals will be, we predict, a very important chunk of customers as well, perhaps more important at some stage.
Okay, thank you. Final question, and absolute time limit. Is it mandatory that you buy your NMR equipment, or you could subcontract this analysis to you?
It would be, I don't know if I understood the final question. It would be important to have our...
You buy your own NMR equipment, correct?
Yes.
Yeah.
Well, initially, in the first year...
Not your customers. You do the analysis for your customers, but you have your own equipment, correct?
Yes.
I think that answers João Oliveira's question.
Yes, I think so.
We now don't have any more time. Well, maybe asking just for one, and it's really the last one. Okay, one here. Bruno Taveira. What are the conditions you can detect? For premature membrane rupture, you have new parameters detected with rapid tests, and it's a much cheaper test. What do you say about this?
Daniela, would you like to answer this? Existing tests. Okay, maybe I can answer that because we are running out of time. So yeah, there are fast tests on the market, but they're still far from ideal in terms of true answers or low rates of false positives and false negatives. So we are aware of those measurements, particularly in blood and particularly aiming at proteins, but they're still not ideal. So PROM is another condition. We are also trying to predict, it's very closely related to PTB, it's one of the origins again. And we are aware that it's important to predict it with more accuracy than it is possible today.
Okay. Thank you very much for your presentation and for your great answers. It is now time for me to jump to the next team. So
okay, thank you very much.
See you in a few moments.
Thank you.
So now it's my turn to call the next team. Okay. The next team focuses in helping us also in diagnosis. This team is called PureDNA, and they aim at helping us speed up the process of disease diagnosis in the field of infectious diseases. To present us this case, we count on Leonor Castro, and the rest of the team is composed of Augusto Pedro, Ana Ferreira, and Inês Cardoso. Leonor, whenever you want, you can start the presentation. Thank you.
Thank you. You can see my screen, right?
Yes.
Okay. Good afternoon, everyone. Today I'm here to present the technology developed by the PureDNA team. As most of us know, infectious diseases are one of the leading causes of global death, according to the World Health Organization, with this number reaching around 40,000 deaths a day. So, if we want to avoid this scenario, we need to perform efficient and accurate diagnosis, and with this, limiting the spread of the disease, reduce the cost of treatment, and more important than that, avoid unnecessary deaths. But you might be wondering, how is this diagnosis performed? Well, currently, after the sample collection, there is a DNA extraction step, followed by an analysis performed by a DNA amplification technique called PCR, which is the gold standard of molecular diagnosis. However, this is the last step, is usually the most time-consuming one due to the low amounts of DNA on the collected samples. So our solution is to increase the amount of DNA on the collected sample, and with this, increase the sensitivity of the PCR while doing faster DNA extractions, and also avoiding false negatives and lower the PCR time. Currently in the market, the extraction of DNA is based on colon and magnetic beads-based technology. But today, the PureDNA team presents their solution based on liquid-liquid extractions. While using these systems, they will be able to extract and purify DNA in one of the phases, while the contaminants will be extracted on the opposite phase. And from this technology emerges the DNA PureTech, which is a DNA purification and concentration kit with vials with pre-filled and ready-to-use mixtures. While using this kit, the user will be able to, after cell lysis, mix their sample within our kit, and after induced phase separation, they will achieve a purified DNA and in one of the phases that can be used in further PCR analysis, and with this, improve the diagnosis of infectious diseases. While using our kit, the user will be able to have already well-established features in the market, such as high levels of DNA purity and also easy integration on lab workflow. But more important than that, they will be able to concentrate the amount of DNA on the collected sample up to 40 times, and they will be able to do all of this in less amount of time and also at a competitive price. Regarding the market that the DNA PureTech will be entering is the global diagnostic market, evaluated in $7.7 billion, from which 1.2 represent the global DNA and RNA extraction kits. This segment can be divided into research and academic centers, pharmaceutical companies, and also hospitals, with our target being 0.60 billions from research centers. Regarding our competitors, the ones that use column-based technology can only concentrate DNA up to 8 times, while the ones that use magnetic beads are not able to concentrate at all. However, using our kit, the user will be able to concentrate the amount of DNA up to 40 times, which is a feature considered very relevant by most of the users of such kits. To achieve all of this, our roadmap is currently on the research and development phase, and we have already established proof for bacterial DNA extraction. So we expect to reach the market by 2022 with an expansion to the USA market by 2026. And to achieve all of this, we will require around 750,000 euros. Regarding the cost of production of our kit, this is estimated to be around 12 euros, and the user will need to spend around 114 euros to use our kit, which is a price that is established not only considering our competitors, but also the features that we are offering. Our sales plan is defined by initially targeting 5 research centers and increase this number up to 3,000 research centers by 2026. And with this, we expect to sell around 100,000 units. And considering the units being sold, we expect to reach break-even after 4 years of being in the market and achieve revenues of around 5 million euros. Last but not least important, the development of this technology was made by a multidisciplinary team from University of Aveiro with a complementary set of skills and scientific backgrounds. More specifically, by Ana and Inês, with a deep knowledge on chemical process and solvents, and by Augusto and me, with a strong expertise on nucleic acids. Moreover, these teams come with the help of scientific advisors, such as Professor João Coutinho and Dr. Mara Freire, which are well-known researchers on the field of liquid-liquid extractions for the purification of several biomolecules. To summarize, the DNA PureTech is a DNA extraction kit developed for research centers who need more and faster and accurate diagnosis of infectious diseases. While using our kit, they will be able to extract and concentrate DNA, but unlike the conventional methodologies, they will be able to increase the sensitivity and reduce the time and costs of the diagnosis of infectious diseases. Thank you all for your kind attention.
Thank you, Leonor. Great presentation, right within the time limit. So now, as in before, we'll have our Q&A. So let's see. I have a couple of questions here. So one of the questions that we have right from the bat is, why did you select research centers as a market entry?
I will let Inês answer that question, if you can.
Hello, good afternoon. I think that the main reason why we think that research centers can be our market entry relies on the fact that we, in fact, have a lot of strong competitors in the field of molecular diagnosis, and in particular in the infectious diseases one. They prevail mostly in hospitals and clinical labs. And taking into account that we are researchers, we think that we can catch the attention of the research center first. And according to our survey, we realize that the majority of the inquired people, which are researchers as well, think that our competitive features are very interesting and relevant. So we think that we can tackle this market first.
Okay, thank you. So now, next question by Ana Fonseca. How does your product solve the problem of false negatives testing?
I guess that Anna can answer that.
Hi. So the false negatives result from a technical difficult associated with the limit of detection of the PCR. So we have samples with low DNA concentrations, and because of that it's difficult to do the quantification of DNA by PCR, and this and because of that we have the false negatives. But with our kit, we are able to concentrate the DNA of the initial samples, and this way we have the concentration above the PCR detection limit. So as Leonor explained during the presentation, our extraction system is composed of two distinct liquid phases, where DNA migrates for one of the phases and the contaminates migrate to the opposite phase. And when we have two liquid phases, then we can reduce the volume of the phase that is rich in DNA, and it's this way that we can manipulate the concentration and obtain a sample with a high concentration of DNA, and this way ensure that we can quantify the DNA by PCR, and we solve the problem of false negatives.
Okay, thank you, Anna. Now, from Bruno Taveira, we have a couple of questions. So, what are the limitations for RNA extraction? What is the plan for automation of extraction? What is the plan for IP protection? What is the plan to target diagnostic centers? So perhaps you should divide among yourselves.
Yes.
To start, I can say that regarding the automation, it would be easy because there are a lot of companies that are specialized in those kind of automatization. So I guess that in that case, it's fine for us. And you have a very simple procedure, so I guess it's fine.
Okay. And regarding the limitations for RNA extraction?
Maybe Augusto can.
I can answer to that question. We are not, we do not see any limitation for RNA extraction. We think that it could be suitable either for the extraction of DNA and RNA, although we do not expect to separate DNA from RNA. In case of co-extraction, we always have the option to add an incubation step with a nuclease, and in that case we can obtain a purified sample of only one class of nucleic acids, DNA or RNA. And the last question was
regarding the question of IP protection.
So maybe I can add something to that. So regarding the IP protection, we have a patent pending, both on the composition of the liquid systems and on the application. And so we have made the provisional patent application, and we're still waiting for the results, but we believe it could be protected.
Okay, thank you. So following, I think we haven't answered, I may be wrong, the one, what is the plan to target the diagnostic centers?
Maybe I can add something regarding that. We intend to reach the research centers first, but we also intend to evolve to other markets. We just thought that it would be good to do the product validation first in research centers, and then we are expecting to evolve not only to other markets, and not only in Portugal, but internationally.
Okay, so another question from Manuel Moreira. Does your technology work with both DNA and RNA? And what about virus and bacteria?
Maybe about bacteria and virus, maybe I can answer. And yes, for now we only have the proof of concept for bacteria, but we believe that our technology can be used also for virus. Since we use a robust lysis method, a chemical one, that allow the isolation of nucleic acids from the distinct source. And the other one, the other question?
I guess Augusto can answer.
Yes. So about the feasibility of using our technology to separate both DNA and RNA, it is possible, but as I said before, we cannot, so let me rephrase. So we can extract both DNA and RNA, but we cannot separate one from each other. So we extract the nucleic acids from non-nucleic acids contaminants, but we cannot separate different classes of nucleic acids. In this case, we can also use a nuclease to obtain a specific class of nucleic acids.
Okay. Another question from Antonio Diniz. Why have you chosen to have a price that is lower than that of inferior competing products? Okay, who's going to answer that?
I'm not sure if I understand very well the question. It's about the price.
Yes, I'll rephrase it. The question is, why did you decide to have a price that is lower than your competing products?
It is not lower, it's in the middle. So I'll try to explain how we get the price. So to determine the final cost of our product, we have in mind three aspects. First, we take in account the kit's production cost, and we take in consideration the materials supply. Since our kits is composed mostly by reagents and vials that are abundant on the market, we managed to set our production cost, that is around 12 euros. Then second, we also have in mind that exists a big competition present in the market, which are well-known companies with a great expertise in the market, like the Thermo Fisher. And third, our position in the market. We considering that our kit have a big advantage in relation to the other ones, that each is the factor of we can concentrate DNA up to 40 times in relation to the initial sample. And so we also know that the kits from our competitors, the price is between 100 until 200 euros. So this way we decide that 115 euros is a competitive cost price for our kit. I don't know if I answer.
I think so. Anyway, we are now out of time. So thank you for your answers. Thank you for the great questions posed by the audience. Keep at it. We'll try to answer them as best as we can. We apologize to those we weren't able to have the questions up here in the air. Anyway, now this team has to go. Thank you very much for the answers and for the presentation. And we're now going to prepare to accept the next team. So next in line is a team called Ceramics for All. This team has a high-performance solution that they think can dramatically change the way armoring is done today. To tell us all about it, we'll have Rui Pinho, and helping him, we have Inês Vilarinho and Ricardo Serrazina. But first, Rui, whenever you're ready, you can start your presentation. Thank you.
Hello, good afternoon. So my name is Rui, and I'm a member of Ceramics for All team. And I'm here today to talk to you about the All for One. So there is a need for armored vehicles, which is related with the protection of the occupants and goods from external threats. To do this, the vehicle needs to be light, so it can be fast and agile, and it needs to be resistant to avoid to be penetrated by projectiles. These projectiles are dependent on the guns that fire them. And this level of protection, 3 and 4, is related with high energy projectiles. To do so, we have two types of materials, metals and ceramics. On one side, metals are very heavy, but they can withstand multiple impacts. On the other side, we have ceramics, which are lighter, but they dissipate energy by shattering. The metals also can be perforated by piercing ammunition. We can compare, for instance, the weight and its effect on the acceleration from 0 to 100 kilometers per hour and time to compare to see the effect on performance. If you armor the Range Rover Sentinel, which is a civilian armored vehicle with a metallic armor, the weight will increase and the time to accelerate, it will be also proportionally affected. If we change for ceramic armor, we can improve the performance of vehicle. Therefore, there is a need for resistant and lighter materials, but also cost-effective solutions. So ceramics are made from powders that are compact and then fired at high temperatures to connect the particles and reduce the space between them. Our technology uses powder engineering and a specific process to reduce the number of pores, meaning the number of the space available between the particles, and strengthen the particle interface. When compared with the conventional technologies, our process is faster and requires lower temperatures, which means that we have lower production costs and we have lower carbon footprint. The Alpha One is an alumina-based ceramic that will be then integrated in defense systems on armored vehicles. The commercial available ceramics will shatter when hit by a bullet. We expect that Alpha One, it will dissipate more elastic energy before breaking. Later, we want to produce the Alpha One composite, which will be the Alpha One, the ceramic plates, incorporated in a defense system, which means it will have absorber plus the ceramic tiles. The absorber is intended to hold the ceramic tiles in place after being heated and shattered. So for the level of armor 4, if you compare the weight and the cost of production for different materials, you can see that steel is it has very low cost, but it's very heavy. On the other hand, ceramics, for instance, alumina, is lighter, but the cost doubles. If we think about other ceramics, for instance, silicon carbide, the weight is slightly decreased, but the cost drastically increases. On the other hand, the Alpha One is more resistant, which means that we can use thinner layers for the same level of protection and still get the job done, while keeping the production costs similar to the steel. The armor materials market was evaluated by 8.5 billion euros in 2017, and it is composed by metals, ceramics, and polymers. Our target market is the armor materials for vehicles. Therefore, it's composed by metals and ceramics, which gives 4.5 billion euros. We will start as ceramic plates producers. We expect to have an equipment partner, DivMac, and we will buy our raw materials and we will sell ceramic plates to defense systems producers. Later, these defense systems will be employed in vehicles, and the final customers will be VIP and goods transportation companies, as well as military forces. Later, with the launch of ceramics Alpha One composites, we will intend to sell directly to producers and modifiers of vehicles. Right now, we are in the research and development phase. We intend to do some patent submission and strengthen our collaboration with DivMac. By 2023, we expect to equip a vehicle, a demonstration vehicle, and in 2024 to do the market entry. And we will expect to sell equip 30 vehicles in Europe. In the year after, we launch the Alpha One composite, and we expect to enter the American markets. then 1 year later expand to Asia and Oceania, and for the later 2 years to be global. The financials reflect the exponential sales. 2024 marks the market entry, and we require an initial investment of 1.4 million euros. The team is composed by myself, Inês and Ricardo. We have 2 scientific advisors from University of Aveiro and 2 business advisors from HiTech program. So to sum up what has been said, for armored vehicle manufacturers who need lighter and high resistance and cost-effective solutions, the All-for-One, unlike other commercial available solutions, is lighter or more resistant by a factor of 2. Thank you for your attention. We will be glad to answer to all your questions. Thank you.
Okay, thank you, Rui. So the rest of the teams will be joining. We have a movie running still.
Yeah, but maybe you can start with the questions. This can simply run if you don't mind.
Sure. So let's start. We have 3 questions now, waiting for more. First question, how can you achieve the enhanced characteristics in your ceramics? This was a question was posed by Manuel Moreira.
Okay, I will ask Ricardo. He is the best person to answer that question.
Thank you, Rui. Hello, everyone. So our ceramic material is produced through a unique technique. And to allow this technique to be employed, we need to change the powders and we need to change the particles that of which we start. So then, while developing the process of sintering, that thing that Rui was talking about, decreasing the space between particles. So as we are decreasing the space between particles, we are also changing the interfaces between them. So this is the reason why we have a material that is more resistant to impact and that, unlike other ceramics, can withstand multiple impacts without shattering.
Okay, thank you. Next, if your material is a ceramic, how do you expect to also tackle the metals market? Are you going to cover only the vehicles market?
So Inês is our market responsible. She can give some light on this.
Yes, so as Ricardo said, our ceramic is more resistant than current materials available. And therefore we can produce thinner plates, ceramic thinner plates. And the market for electric vehicles is increasing. So weight is very important because nowadays you cannot armor a vehicle and in both at the same time have an electrical vehicle. So that's why we think that our material can tackle this market. About the applications, we expect in 5 years' time also to have the personal vests market, and in 15-year times, more or less, also the drones.
Okay. So, a question from across the ocean from a mentor from HiTech One, Angus Kingham, also is placing a question to you. Did you consider the personal protective armor market, example, armor vests for police forces and the military? He thinks that it is a larger market and possibly easier to enter than the vehicle market. Or is it both? What do you think?
So I can answer. So the armor vests actually are not the higher market. The vehicle armor is the highest market in this protective, with this protection and for these materials. So that's why we started with the vehicles, and then we will go to the vests market and armor market. Also because the armor materials for vests are also more difficult to enter in the market. So we started with the vehicles and then we will go to the vests market.
Okay. So now from José Borges, a series of questions. How does your solution compare with new materials such as the auxetic materials, which are lighter? Do you want me to go one by one, or do you want to hear all of them in a row?
You can go one by one.
Okay, so this one.
Okay, the auxetic materials are used mainly on body protection and helmets. And as the audience noticed, we left Kevlar out of our presentation, but it's also armor material. It's just not used in vehicles because it's used for a lower level of protection. Okay, and these auxetic materials, they are lighter, but they are in the lower side of the scale. We are aiming for high level of protection. Therefore, the level 3 and 4 to avoid piercing ammunition and high power projectiles.
Okay, so the next two ones I will join because I think they're related. Where did you perform the testing for your technology, and did you go for the certification process?
So for the testing of our technology, actually we have contacts in Army, and the Army is open to test our materials. So as soon as we have a proper size sample, they are able to test our materials right now. For the certifications, we are already looking for independent laboratories that can run these tests and deliver this kind of certification of the material.
Okay, thank you. So now let's go for another one from a former participant in the program. So Francisco Rayo asks, you showed projected sales as a number of vehicles sold. Do you intend to produce and sell the vehicles, or will others do it? Are you going to measure sales in vehicles sold or by weight of material produced and sold?
Okay, I will ask Ricardo to answer to this question. I think he's...
Sure. So in this case, in order to make our math simpler, we, in terms of market and in terms of sales, we considered an average area to be armored in a vehicle. So we took several vehicles that need armor, for instance, the vans for goods transportation, the light vehicles on the Army, or the Range Rover Sentinel, for instance. We calculated an average area that needs to be protected, that needs to be armored, and then we consider a constant thickness all over this area. So by this, we calculate the number of tiles, and we calculate the number of vehicles that we want to, or that we will tackle and armor throughout during our market entry. So to answer you, of course, we will consider selling the material, kilos of material or something like that. But to make things easier here to explain, we consider vehicles and we consider a mean area for vehicle armor.
Okay, thank you, Ricardo. And saying thank you, team. We are now out of time for more questions.
Thank you.
Thank you for the great answers and for the presentation. I'll apologize for the remaining questions. Please don't quit. Keep sending questions. We'll try to answer them as soon as we can. Guys, we'll maybe later on try to answer them as well offline. Okay?
Thank you.
Thank you. Now it's time for the next team. And next, we have a team that is proposing to help cure our broken hearts. No, it's not a dating app. What we have here is a solution to help save lives by fixing some well-known problems with hearts. And to tell us all about it, we have the GOTech team, who is composed by André Pereira, Helena Ferreira, Inês Gonçalves, and Patricia Henriques, who is going to tell us everything about this project. Yes, Patricia?
Yes.
Start whenever you want.
Okay. Okay, so good afternoon, everyone. My name is Patricia, and I'm part of GOTech Cardiovascular, and I'm here to present you our medical device that can mend your broken heart. And why do you need this? Well, it's not because your love life is miserable, but because just during the time of this speech, 4 people will have died of coronary heart disease. This disease occurs when the blood vessels from the heart block. To solve this, surgeons perform a bypass surgery to re-establish the blood flow in the heart, and for that they use other vessels that are called grafts. Every year, around 1 million surgeries are performed worldwide. So the gold standard in this procedure is to use autologous grafts, meaning vessels that are harvested from the patient himself, even though they are not always available, and the harvesting is usually associated with comorbidities such as infection. Still, since there's no other solution, surgeons continue to use them. So the mammary arteries are the preferred options due to their good performance. However, here comes trouble because we only have 2. And in general, most patients need at least 3 grafts. So for the remaining ones, we need to go for the secondary options, which are generally associated with high failure, in some cases reaching 50%, and mostly due to thrombosis. When this happens, this leads to the occlusion of the vessels again, and patients end up needing a second intervention, or they actually die. So I'm sure you agree with me that there's an urgent need for vessel substitutes that are readily available, prevent thrombosis, and are a long-lasting solution. This need has been validated by cardiothoracic surgeons and also by the fact that synthetic grafts have been explored for coronary artery bypass, but they have failed, and once again, due to thrombosis. But fear not, because we have the solution. So we've developed a technology based on polyhema, an FDA-approved hydrogel that has anti-adhesive properties, meaning that it does not prevent the adhesion of anything onto its surface. However, since it's an hydrogel, it's mechanically weak. And this is where we had our eureka moment and decided to incorporate graphene, the strongest material on Earth, in order to obtain graphema. So graphema is a strong hydrogel that still is able to maintain the anti-adhesive properties of polyhema. We have shown this in vitro, where we see there's no human platelets, bacteria, or cells attached onto its surface. This is important to prevent thrombosis and infection, and so this material is promising for the use in the development of materials for contact applications. And that's why we have used this technology to develop our first product, the GoGraft, which is a synthetic small diameter vascular graft that prevents thrombosis and infection. We tested it in pigs, and we compared it with a commercially available material for vascular access. And what we saw was that there were no thrombus formation on the surface of our material, as contrarily to what we saw in the surface of the other materials. In addition, when we use GoGraft, it's readily available at the beginning of the surgery, and it has a long shelf life. Using GoGraft also brings other benefits for both the patients and the healthcare system. So if we avoid the vessel harvesting, we manage to reduce the recovery time and also associated infection risk, and also reduce the hospitalization and surgery time, releasing the operating rooms. Apart from saving lives, this also has a huge economic impact for the healthcare system. So if in each bypass surgery, we replace the use of autologous vessels by the GoGraft, we can achieve a reduction of 13% in the procedure cost, reaching a potential savings of 2.83 billion euros. In addition, if we also manage to avoid the need for that second intervention, we can add up another 0.54 billion euros, reaching a potential global savings of 3.37 billion euros. So our GOTech Cardiovascular would be inserted in the vascular graft industry. So we would receive our raw materials to use in our R&D department that would be focused on the development of GoGraft and other products. We will then outsource our large-scale production, sterilization, and packaging to a contract manufacturer, even though we'll be ourselves performing the quality assurance. And then we will sell our products to distributors with well-established distribution channels and with privileged access to both the hospitals and the cardiothoracic surgeons. To improve the visibility of the product and the confidence of the surgeons, we'll be also performing medical training. So our sales and marketing strategy will focus on the countries where we have an IP protection. So after finalizing the product development and the clinical trials, we'll enter the market in 2029 in Portugal, of course, and in UK, France, and Germany, considering their high number of bypass surgeries and high economic power. In the following year, we expect to expand to the remaining European countries and by 2031 to reach the US market. So after 10 years in the market, we expect to be selling around 900,000 units per year. The GoGraft production cost is estimated to be around 300 euros, and so we expect to sell our product by 1,000 euros in Europe and 2,000 euros in the US, with a gross margin between 70 and 85%. To get the product to the market, we still need to work on the final product design and perform some long-term in vivo studies, and for that we'll need some investment. We'll also need to perform the preclinical studies already in GLP conditions and the clinical trials. We'll start with an early feasibility trial using 10 to 15 patients and then expand to a larger pivotal trial for which we also be needing the largest round of investment before we get into the market. During all this time, portfolio expansion will continue. So balancing all of these, we expect to reach our break-even 1 year after the market entry and 4 years later to reach a net profit of 261 million euros. So we're researchers at I3S, the biggest institute of health in Portugal, and we have expertise in the development of materials for blood contacting applications. To summarize, for cardiothoracic surgeons who need vessel substitutes, our GoGraft is a synthetic vascular graft that is readily available and prevents thrombosis and infection. Unlike vessels that are harvested from the patient, our GoGraft is ready to use in a one-step procedure, reducing the surgery time. So thank you.
Thank you. Great presentation. So let's start now with our Q&A. First, by Bruno Taveira, can the GoGraft or the coating be used for other medical devices like catheters?
Yes, so maybe Andrei can answer this question.
Okay, thank you for the question. We actually filed the application patent for our technology and also that protects also our product, that protects the production methods and also several medical applications. As graphema is anti-adhesive hydrogel and is also mechanically strong, it has features that could be applied for blood contacting devices, such as the catheters that Bruno Taveira mentioned. And besides this, we can also apply our technology for the design of heart valves and also for other medical applications, such as cartilage and intervertebral disc. So as our product, the GoGraft, besides the application for coronary artery bypass grafting, we can also advise its application as a product for peripheral artery bypass in the legs and also for vascular access for dialysis patients. So this opens a lot our portfolio of applications.
Okay, and still from Bruno Taveira, do you plan to license the technology to the big medical device producers?
Ines, you want to address this one?
Yes, I can answer this one. Thank you, Patricia. So we designed this study using us as the producers now when taking it to the end. But we do think we would have advantage of licensing the technology to a medical company that already has all the procedures and the fabrication methods running, because we do need to have different licenses to produce the medical products, and so they would already be used if it's a similar company producing the catheters or any vascular grafts. They would already have all the technologies and the standards designed for this. So I think we would have the advantage of licensing it.
Okay, thank you. Next question from Joanna. What is the worst scenario for your business plan since you are so far apart for market introduction? Maybe when you get there, there will be similar technologies available. Ooh, challenge.
Ines or Elena, one of you want to answer?
Elena, you want to go? Yeah. So I guess that we are far from the market, but maybe Elena can then tell you some insights on the long way run, because actually we are far away because we do need to do some clinical trials on this, because it's a medical grade device, so we really need to do the in vivo studies. But actually, in the worst case scenario, so if we would not be able to enter as the Go graft itself, we would really have a big advantage of using it as a catheter, because the product is already designed as it is for use as a catheter. But we actually chose to go into the small diameter vascular grafts because there are really no products on the markets, and in the past decades, no innovation has been shown. And so we think it's a really pressing need. But maybe Elena can just give a small highlights on the path of the regulation, so that you understand why we need so many years.
Yeah, I mean, that basically, I think, answered the question, but just to add up, so yeah, as we mentioned in the presentation, we have to go through basically sort of 2 stages of clinical trials, the early feasibility, which is a small study with 10 to 15 patients, and then a pivotal trial with about 1,000 to 1,500 patients. So bad news is there's a long way to go there, although the need is extremely important to be solved. Good news is anyone who does a product like this has to do this sort of trials. So there's no other way to go around the question, because it's a high-risk medical device. It's classified, it's a class 3 medical device. So any sort of competitor who also wants to get into this market segment has to go through the same clinical trials. That we know of, there are no other companies working on something similar. It's something to take into account, and it's important to look at the market and keep an eye on it. But yeah, I think we also have backup plans, as Inês mentioned. So.
Okay. So would you say that the currently available materials available in the market for vascular accesses would be your competitors, or not at all?
Inês, you want to answer this one?
Okay, so no, so the currently available materials are the EPTFE or the Dacron that are available for to be used as grafts for medium and large diameter vessels. But in terms of, and they work well, but when we go to small diameter vessels like the coronary arteries that we want to tackle, they don't work, so they clot. So they are not really our direct competitors because they cannot be used for the coronary arteries. And therefore the only direct competition that we have, it's really the autologous grafts that have all the problems that we mentioned during the presentation.
Okay, thank you, Inês. We now are out of time. It's the story of this session. So thank you for your answers. Thank you everyone for the questions that you put in. We are now reaching the moment for our break, so 10-minute break. So I'll thank the team. Thank you very much for everything. Thank you for the presentation and the Q&A. And to the rest of you out there tuning in, let's grab a small 10 minutes break to rest our eyes and ears for more teams to come. Okay, 10 minutes, don't be late. Hello, everyone. Shall we start again? So it's now time to resume our session. Hope everyone is live and ready. Okay, I think so. So let's start. The second row of teams is going to be initiated by a team aiming to solve a big problem that all of us hear about: the issue of cancer. This team, aptly called Chemi-Tumor, is composed by Luis Silva, Diana Pista, Patricia Berdulas, Sonia Fernandes, and Carla Magalhães. I'll ask Carla to present this team's work and excellent project that is a true challenge for cancer. Carla, whenever you want, you can start.
Good afternoon, everyone. My name is Carla Magalhães from Chemi-Tumor, and I'm here to present you our product, self-activated and cancer cell-specific drugs. I'm sure all of you have heard of this terrible disease, cancer. In fact, this disease is the second leading cause of death all over the world, and it caused almost 10 million deaths in 2018. At the time of diagnosis, half of the patients present metastases, and for many years, people have been trying to treat this disease with chemotherapy without success, once this therapy is ineffective in the targeting of the tumor cells and causes severe side effects to the patients. In the last years, a lot of biological therapies have been developed as target therapy and immunotherapy. However, these therapies only can be applied to a specific subset of patients and still have some limitations. So our team have a revolutionary approach for cancer treatment in which we can target only the cancer cells. And how can we do that? We have found a cancer marker, the superoxide anion, and based on that, we have created a new set of small molecules that can be applied to all cancer patients. After administration, these compounds will reach the cancer cells, being activated by superoxide anion, leading to reactive oxygen species release. These species are responsible for tumoral cell death. This technology is already under a patent process. We have tested this technology in several cancer cell lines as prostate, gastric, breast, liver, and lung cancer. And as we can see here, with the increasing concentration of our compounds, there is a decrease in the number of the cancer cells. When tested in healthy cells, these compounds have no effect. So we can conclude that this technology has a broad applicability for the treatment of different tumors, being safe for healthy cells. We decided to apply this technology in the most incident and deadly cancer, the lung cancer. The most incident type of lung cancer is non-small cell lung cancer that is classified in 4 stages. Stages 1 and 2 are localized, and the first-line treatment is surgery, with which we cannot compete. Stages 3 and 4 are metastatic, and the treatments are ineffective, and the survival rate for stage 3 is 32%, and for stage 4 is only 6%. Besides, there are some more aggressive cases that are called unstaged, and currently the patients don't have a therapeutic option and only receive palliative treatment. So our goal is to apply this technology as a breakthrough therapy for the unstaged non-small cell lung cancer patients that don't have therapeutic options, so we can get a faster approval of this therapy. After we prove the good performance of this therapy, it can be applied to stages 3 and 4 of non-small cell lung cancer and eventually to the other cancer types that we have already proved the anti-tumor effect. In this process, our team will be responsible for the research and development and the quality assurance. The preclinical development and the product development will be outsourced to CROs. After these processes are concluded, we will license our technology to pharmaceutical companies that will be responsible for the clinical trials and the distribution of this product to the hospitals. In order to estimate the licensing value of our technology, we have created a market model in which we are considering the progressive entry of this therapy for the stages of non-small cell lung cancer, and we have defined the pricing point in 10,000 euros per patient a year. Based on that, we have obtained the price for the licensing of 500 million euros with an upfront fee of 30 million euros. These deals have been made for other non-small cell lung cancer therapies as pembrolizumab and lazertinib, being that lazertinib was licensed in $1.25 billion. We have already attracted a part of the funding for the study of mechanism of action, in which we are working currently. The next steps of preclinical development and product development, we need an additional fund of 3 million euros, and we expect for these processes a period of 3 years. After these processes are concluded, we will license our technology before the clinical trials, and we expect a period for the clinical trials of 5 years with an investment of 40 million euros. Here is the team that will make all of this happen. Luis Pinto, an expert in luminescence and physical chemistry, me in biochemistry and oncology, Patricia Berg-Julius, an expert in synthesis and medicinal chemistry, and Diana Christian Sonia Fernandes, making the connection with the industry and giving us knowledge in chemistry and biology. So our team have a revolutionary approach for cancer treatment in which we can target the cancer cells while being safe for healthy cells. Besides, this technology can be applied as a therapy for all cancer types. Thank you for your attention.
Okay, thank you for your presentation. We're now going for the question part. Are you ready, Tim? So, first question. What can you tell us a little bit more about the licensing cost that you presented? This question is from Ana Fonseca.
Okay. So we have created a model to estimate the incomes of applying this therapy, and we have calculated based on percentages our license cost. Besides, it is a smaller license cost once we want to license before the clinical trials.
Okay, Carol. By the way, this connects with Antonio Dini's question, which is asking, so your idea is to license before the phase 1 clinical trials?
Yes, if we can do that, we want to license before the clinical trials.
Okay, so additional question from Manuel Moreira. What is the chemical nature of your compounds? What is the process the compounds use to specifically target cancer cells?
So, to target cancer cells, our compounds will reach cancer cells and healthy cells, but in cancer cells, there is this cancer marker, the superoxide anion, that will be responsible for the generation of reactive oxygen species that will kill the cancer cells. About the nature of the compounds, Patricia can tell more about this.
Our compounds are small molecules. Their structures are based on the structure of cholinteracin, which is a natural compound, and basically we have two aromatic fragments joined together. It's a very simple molecule. It's a flat molecule and can be synthesized in very few steps.
Okay, thank you. Now from João Silva. Thank you for your presentation. You have mentioned the inexistence of therapies for the type of cancer you are targeting. On that basis, have you considered to request for an orphan drug designation?
Yes, we consider that, but we think that it's easier to apply this therapy for the end-stage non-small cell lung cancer, as I told you, because the patients only receive palliative treatment, so we can get a faster approval for this cancer type. As an orphan drug, it is a possibility for another cancer types, but for lung cancer, it makes sense the end-stage cases.
Okay. Thank you. Felipe Castro is asking for what are your plans for preclinical trials?
Maybe Patricia can answer that.
We plan to start as soon as possible with the in vivo development, with the preclinical stage of our products, our drugs. And our intention is to establish some partnerships to advance in this stage. And when we reach the point at which we cannot do any more from academy, we plan on getting some funding to outsource the rest of the preclinical trials and the dossier and everything.
Okay, thank you, Patricia. Question from Antonio Diniz. The 10,000 euros per treatment, is that what you will get from the licensor or the price of the drug?
Yes, the 10,000 is the price of the drug, and we use that value to estimate the price of the licensing. It's not the price that we will obtain, it's the price of selling the drug.
Okay. And this is a happy coincidence. We've reached our time and the questions. So this is a new record. Thank you.
Thank you.
So thank you, team. Great Q&A, great presentation. It's now time for the next team to show up. So kindly see you in a few.
Thank you.
Okay. And we'll now transition to the next team. This team, it's called Phyergy, and they aim at looking in a different way to two big problems that we deal with nowadays. The issue of waste, or more specifically food-related waste, and the issue of energy production. So this team is composed by Ivo Pimentel and Vitor Cândido, and Ivo is going to tell us all about this new approach to turning waste into energy. Ivo, whenever you want, please share your screen and start the presentation.
Hello, good afternoon. My name is Ivo. I'm one of the founders of Phyergy, and I'm here today to present you our disruptive waste to energy technology. Climate change and the way we use our limited resources have created an unprecedented challenge. In Europe, 90 million tons of food waste are generated every year. And just to move this waste from where it is produced to centralized treatment facilities, we need to travel around 2 billion kilometers. Most of this waste ends up in a landfill, which has a huge environmental impact. And additionally, still today, most of our energy is coming from fossil fuels. So Europe has ambitious objectives to transition to a carbon neutral and a zero waste society. And these systems are outdated, extremely inefficient, and just ready to be disrupted. So we're looking at different technologies to address these problems, and we found that anaerobic digestion is recognized as the most efficient way to convert organic waste into carbon neutral fuel. On the other side, we have the hydrogen fuel cells that are an emerging and disruptive technology. They produce energy through a series of chemical reactions, have no moving parts, and are really the most efficient zero-emission technology available today. So by combining an available and carbon-neutral fuel with an emerging zero-emission technology, we have created a cost-competitive and unbeatable carbon reduction solution that we have called the biopod. So the biopod is disrupted because it is installed where the waste is produced, eliminating the need for waste movement and landfills. In this way, waste is also valorized into zero-emission energy. The biopod is packaged in a plug-and-play containerized solution, fully digital with IoT sensors for cloud monitoring and preventive maintenance, and is able to process the food waste of hypermarkets, for example. So we are extremely excited about the biopod because we see it applied everywhere, in city parks, in schools, hospitals, farms, or supermarkets. The biopod in the future will be integrated into the buildings or even on the streets to serve communities or neighborhoods. We are planning to do our demonstration project with a supermarket chain that will lead to a long-term contract to decarbonize their stores. We have already validated the interest of the biopod with the largest supermarket chain in Portugal. And just last week, Tesco announced they want to become carbon neutral by 2035. So the biopod addresses the needs and requirements of the food value chain from production to consumption. The biopod has a target price of 280,000 euros with an interesting 40% margin and with further cost reduction potential as we scale up. It eliminates the rising costs of waste disposal, reduces your energy bill with a profitable return on investment and with a payback within 6 years. Additionally, it will boost the social and environmental responsibility, improving resilience and efficiency. So we have considered the countries that present the most favorable market conditions and are historically more open for innovation and environmental solutions. Selected countries have also the highest costs for the main value drivers of the biopod, and also have in place subsidy schemes for these kind of technologies. It also happens that most of these countries are the biggest European markets. So to solve the food waste problem in Europe, the market value is estimated at 64 billion euros, 45 billion for the selected countries. And if we are just looking at hypermarkets in these selected countries, the market value is around 2 billion euros. So in terms of competitors, there are two companies offering decentralized and small scales anaerobic digestion solutions in Europe. And as for the fuel cell industry, it is still small and highly fragmented. There are a handful of fuel cell integrators in Europe, and the solutions available today are either too small or too big for the biopod. So there is a bottleneck in terms of fuel cell to apply it in containerized solutions. And this is where our expertise and know-how in the hydrogen economy and technologies gives us a competitive advantage. So we are looking for a seed round investment of 400,000 euros that will take us through three main milestones. put together a top-notch international team, build a smaller, fully functional prototype as a proof of concept, and complete the biopod concept design. And this should be enough traction to open round A that will take us through the biopod development and up to the demonstration projects. First sales should happen around year 3. That will open also the round B of investment to scale up the company. Factoring in all required capital investments and operational expenses, we expect to achieve the break-even around year 5. By year 6, 7, we expect financial independence and growth that will support our planned R&D pipeline and market expansion. We also expect around this time to hit the 1,000 unit sales milestone and have around 150 people employed. So Vitor and myself, we have been working on and off for more than 5 years and are partners in other ventures. I am the co-founder of a successful Munich startup working with hydrogen technologies for the automotive industry. It was actually selected one of the top 100 energy startups this year. And Vitor brings all his management, entrepreneurial, and sales experience to the company. We are already scouting for the core team and the board of advisors. So a society needs to reinvent itself and we need to do it fast. In the cities of the future, there is no waste, but resources. They are fully circular, connected, and clean. So thank you for your time and attention.
Okay, thank you, Ivo. It's now time to remove the screen sharing, please. And meanwhile, we have a couple of questions here. So, first question out of that, why did you choose hypermarkets as an entry market?
We realize that retail is a very competitive market in attracting and maintaining customers and to get some brand differentiation. And as we all notice, the investment in solar energy by these companies to decarbonize part of the operation is happening. At this moment, we are with a problem at the table by circularity and zero society about organic waste problem. And that's why we really believe that biopod can solve locally this issue by closing the circle and transforming bio-waste into a value asset like energy or heat.
Okay, thank you. A question from Angus Kingman from the other side of the ocean. What is the composition of the resultant gas of the anaerobic digester? Is it ideal for the fuel cell?
What is coming out, what the microbes digest the food waste and they convert it into biogas, which is mainly methane and carbon. So this is actually a perfect fuel for the fuel cell because the fuel cell can reform internally the biogas. It converts the biogas into hydrogen, which then feeds the fuel cell. So the fuel cell is fully ready to take biogas or actually other fuels.
Okay, thank you. Question from Francisco Raio. How many biopods will the average supermarket need? Will one biopod per supermarket be enough?
Yeah, I mean, the biopod has the capability to convert 1,000 kilograms of food waste per day, which is a perfect fit for hypermarkets. So, yeah, I mean, it would solve their waste problem, let's say.
Okay. A question from Manuel Moreira. What is the technological uniqueness of the biopod as you are just combining two existing technologies? So how can you protect it?
True. I mean, as many other solutions that sometimes come into markets, I mean, like electric vehicles, for example, I mean, you are combining batteries into already existing car, which has wheels. And so in reality, we are, let's say, taking advantage of all our know-how and expertise within the hydrogen technologies. And finding there is a problem to be solved, and we are combining these technologies. Now, since we are breaking ground here and the solution is new, and we expect that in the future we will see much more decentralized, let's say, solutions of this kind, there's definitely a lot of IP to look when we start putting our hands on engineering.
Okay. Another question from Pulsar. What has to be done crucial to take the hurdle of providing the first demonstrator in terms of most important actions, partners to pick, competencies, strategic partners? What do you think?
Yeah, I mean, so as any other startup, this is a roller coaster, so there's a lot of work to be done. First of all, we need to have a really good team, right? So we are aiming for an international team with experts from the fuel cell industry, for example. I mean, so what we really want to do is we need to actually find the suppliers for the fuel cell stack. So we want to buy kind of off the shelf the fuel cell stack, but we want to develop in-house all the engineering about the combined heat and power and balance of plants around the fuel cell, right? And of course, we are entering into let's say, an emerging technology, which is technology and solutions, which is the hydrogen economy. So we need to establish ourselves within the industry, right? So yeah, I mean, I would say that those are probably the biggest challenge we need to face at this point.
Okay. Next, from Katia Mago. What is the H2 purity produced by the fuel cells? I assume the hydrogen purity produced by the fuel cells.
That's a good question. I mean, the kind of fuel cell that we have selected does not require high purity, like PEM for the automotive. So it actually reduces costs also.
Okay. So last question from Rui Rosas. Is it your business model considering exclusively a forward-front CapEx investment from the customer, or are you considering alternative models?
Yes, we are considering alternative models. I mean, these are more complex. We need more time to understand it and study it, but we're definitely looking into what could possibly be kind of a disruptive business model. I mean, for now, it's CapEx investments. It's what we have on the table, but we're definitely developing different approaches to the business model.
Okay. Thank you, guys. Guess what I'm going to say now? We're out of time. So thank you for the presentation.
Thank you very much for questions.
Yeah, thank the audience for the questions. Great questions. We still have a few, so probably you can try to answer them through offline means. But for now, we'll need to move on to the next team. Okay?
Okay.
Thank you very much, Ivo. And
thank you. Thank you, guys.
Now we are going into the final team. Last, not the least, and a great project, we have the Diagonal team. This team is aiming to solve key challenges in the pharmaceutical manufacturing processes. They aim at significantly enhancing the process and the flow of the existing process in a specific applications. But to tell us all about it, we'll count with Ana Catarina Souza, but we also count with the rest of the team for the Q&A later, Ana Rufino, João Valente, and Ramiro Pastorini. For now, Ana, whenever you want, please start sharing your screen and start whenever you like. Thank you.
Can you see my presentation now?
Yes, we can see it. Just need to put it.
Just trying to. Okay.
Thank you.
Can everyone see me and hear me properly?
Yes.
Thank you, Olímpio, so much. So we are the last team of this day. My name is Ana Souza, and I'm here to present Diagonal team that develops MARDG, a new technology to reduce the costs of biopharmaceuticals. Monoclonal antibodies, or MABs, are one of the most powerful tools in modern medicine. They are used in untreatable or hard-to-treat diseases, and that is why, over the last 30 years, more than 150 MABs has been licensed. They have high demand. In 2019, for example, 11 of the 25 top-selling drugs were MABs. Furthermore, it is expected that this demand will increase in the future. In fact, the access to monoclonal antibody-based products was recently considered as a global health priority. So in order to be able to respond to this increasing demand, the pharma industry invested in the bioprocessing. This bioprocessing is very complex, involving two major steps: the production, or upstream, and the purification in the downstream. Up until now, the investments made streamlined the upstream steps, allowing the production of large amount of MABs, being the main bottleneck to downstream, which accounts for over 60% of the bioprocessing costs. At this stage, we need to extract the MAB from the very complex medium that comes out of the upstream that includes, for example, the feedstock, cell secretions, and other proteins. So in order to maximize the entire process, improvements in the purification steps are necessary. Currently, the purification involves a chain of highly sophisticated techniques, starting with the clarification and then the key step with definitive chromatography with protein A, which is biological based, very sensitive and expensive. With this technique, the target MAB binds to the Pro A in the resin, then we need to wash to remove the impurities, and then we need to elute the MAB by using a buffer, generally at low pH, which can affect the MAB stability and the lifetime of the column. And this purification steps requires multiple repetitions because even the best columns cannot get all the MABs in one go. And the worst part is that for some hard to purify MABs, this approach is not even efficient. So there is the need to develop new strategies to meet these challenges in an innovative way. And this is exactly what we are going to do. We will improve the downstream by simplifying the process and replacing the critical steps, including chromatography. But how? Well, we will use a well-established technique, liquid-liquid extraction. The innovation is that we will use designer solvents that we will fine-tune for the specific biopharmaceutical. These solvents can be used to create aqueous biphasic systems that will allow to separate the target MAB into one of the phases in a single step and at neutral pH. We have proven that this is possible for several target compounds, including proteins. We have several scientific publications on this and one patent. We are now developing the patent for MABs. In the industrial pipeline, we will substitute the clarification and the critical Pro A by one single step. This means that the soup that comes from the upstream can be directly used, which allows to streamline the process. We will also substitute the other chromatographic steps. Our technology uses solvents that are non-volatile, and the extraction is water-based, and thus the entire process is more sustainable. We can also reduce the time and increase yield given the higher capacity separation of our method. Furthermore, MARDG opens the door to continuous mode of operation, something the industry really wants. We are now in the process of setting up a collaboration with AstraZeneca for the purification of a specific MAB sample. Our main competitors are here identified, and they provide the traditional Pro A columns. We also have the competition from emerging technologies like alternative ligands to Pro A and membranes. However, they are still very expensive and do not allow the single step. Our market is the antibody bioprocessing market, evaluated in $13.3 billion. But because this market comprises the entire bioprocessing and we are only addressing the purification steps, our addressable market will be about $8 billion by next year. We aim to enter the European and North American markets, where most companies that need to streamline the downstream processes are, either biopharma that have the MAPS production facilities or the contract manufacturing organizations. In our business model, the client will provide us the culture medium with the map they need to purify. Our knowledge-based approach will allow us to fine-tune our platform to this specific map and then to develop an efficient, sustainable, specific purification platform for this biopharmaceutical. Together with the client, we will work on the IP and we will license the method of use. For the financials, we consider that the general map is about 26 million euros and that the purification steps account for 60%. So considering that our technology can improve the yield by 20%, the client will have 3.1 million extra profit per year. We will divide this extra profit with the client, which translates to 6% royalties, which is the industry standard. We will receive upfront fees for the fine tuning. We will license our first MOU in 2024, and after 4 years, we will reach a net income of 20 million euros and in 2034 of 15 billion euros. In our road to success, we have already developed the purification technology at a lab scale, and we can successfully purify MAPS, and we are working on the IP protection now. We need 3 more years and an investment of 1.5 million euros for the optimization and fine tuning. We will work together with our partners in the industry in order to develop specific solutions for their most pressing needs, including not only MAPS, but other biopharmaceuticals, like, for example, cell and gene therapy or bispecifics. We will do this because we are a highly motivated and multidisciplinary team, deeply engaged in this project with more than 30 years of combined expertise in biological sciences and more than 10 years expertise in protein purification. So to sum up, MARDG technology for monoclonal antibodies purification is for the biopharma industry that needs to streamline protein purification. Our platform, unlike other conventional techniques, is highly adaptable, improves yield, reduces time, and is more sustainable. And thank you for your attention.
Thank you. Okay, thank you. We're now live. So final round of questions for the afternoon. Are you ready?
Yes.
Okay. So how certain are you that the biopharmaceutical industry that is very much risk adverse will switch from an established purification process to a brand new one? Have you validated the industry pain you are willing to address?
Okay, thank you so much. For, sorry?
From Manuel Moreira.
Okay, thank you, Manuel Moreira. Thank you for the question. It's really important one. Yes, we have validated this with the industry. We talked to many different partners that have this pressing need, and they were very excited with our alternative method. We are now working with one of them. Well, we mentioned that during the presentation. I don't know if my colleagues want to add anything for this.
Just let me add something. The thing is that the industry is still struggling with bi and tri-specific monoclonal antibodies, and which they cannot purify at all. So they are much open to new strategies and technologies that will actually allow the purification of those kind of monoclonal antibodies.
Okay. There are so many biopharmaceuticals that they want to purify, but they are still struggling, and we can be of help for those biopharmaceuticals that they are still struggling.
Okay. Next from Joanna. What is the technology behind the tune specificity you provide? It involves other antibodies or is only chemical?
Okay, thank you so much for the question. I will pass to Ana Rufino to answer this because she's our specialist in the lab. she had the hands-on on everything that happens there. Anna?
Yes, so thank you for the question. We only need to prepare the system, water, and a combination of polymer, salt, or ionic liquids. We don't use another biomolecules to the preparation of the system.
Okay. Fast.
So non-biological based.
Okay, thank you. Next question from Ana Fonseca. How can Diagonal team help the pharma industry to recover their investment?
Okay, maybe this one, Ramiro, again?
Okay.
Or João?
The thing is, of course, the IP protection is a very good thing because you have your intellectual property limiting the use of a molecule that you developed and that you purified. But at the same time, this comes as a curse, if you want, because you have limited time to basically help recover the investments. By expediting and speeding the process of purification, actually we give the industry the power to recover their investments and then maintain the sustainability, in the investments of chemical innovation.
Thank you. Now from Patricia Rodrigues. How do the results you get so far compare to those obtained by protein A chromatography?
Okay, again, I will pass this to Anna, the girl in the lab working with the samples.
So we can compare our results at lab scale with the Pro-A performance because we can obtain yields and purity above 95%, and we can increase these values and we reduce the steps comparing with the traditional steps using in the industry.
Okay. Final question. If I were to give you 1.5 million, what would you get? Or what would you give?
First, thank you for your generosity. But then our man of the financials is João Valente. João, can you please answer this one?
Yes, thank you very much for the question. From the roadmap, it is shown that the 1.5 million is complete to research and development, and we can reach the market after 3 years. If you gave us that money, right now we can address that money to have more resources and to prove the performance of our products. So we can reach the marker sooner, about 6 to 12 months. For you, this means that you have a value asset in the company that has shown results in performance, and you can have your money back after 4 years of the product enter the market, just from upfront fees.
Yeah, so everyone wins, Alipio.
So give us the money, please.
Sorry, I'm just the middleman.
Do you take the 10%?
Let's go for that. That's enough. With your numbers? Okay. Anyway, thank you guys. Thanks to everyone watching. This was a great afternoon, ended with a great presentation and a good Q&A. Let's go now for the finishing of the session. Team, thank you very much.
Thank you.
Thank you.
Congratulations to all.
Congratulations to all teams, guys. It was great.
Hey, you're making use of the fact you're last. Come on.
Of course.
Bye. Thank you.
Thank you.
Bye.
Thank you.
Okay. Now we are reaching that moment of saying goodbye, but not yet. We still have someone else to give us finishing speech. To help us close this session, to help us round this afternoon communication and experience, we are now going to have Mario Freire, Vice President of HiTech Association, talk to us and tell us the association view on this afternoon. Mario,
many thanks, Alipio.
Seria o Presidente da Associação, António Bradão de Vasconcelos, a fazer o discurso de encerramento. No entanto, por um motivo imprevisto pessoal, ele não pôde estar presente. Assim, por sua solicitação, ele a ler em seu nome o discurso que previa dizer, agradeceu a presença a todos os intervenientes. Excelentíssimos senhores dirigentes e quadros das empresas associadas da HiTech, excelentíssimos participantes do programa HiTech, excelentíssimos senhores e meus senhores. As minhas primeiras palavras são, naturalmente, de saudação às pessoas que participaram nesta sessão de encerramento do programa HiTech e, em particular, ao que neles estiveram envolvidos ao longo dos últimos meses. Um agradecimento especial é devido a quem nos apoia nesta missão. À equipa da HiTech que torna a concretização deste programa possível. Aos 25 investigadores que apresentaram os seus projetos de negócio, aos 7 mentores que acreditam no programa e de forma voluntária apoiaram de forma incondicional as equipas, ao Roger Debo, da Universidade de Rutgers, que mesmo com a atual conjuntura disponibilizou o seu tempo para apoiar as equipas nesta reta final do programa, aos associados da HiTech que acreditam no valor acrescentado deste programa para a sociedade e que, por isso, investem financeiramente no apoio à investigação, à tecnologia, assumindo um compromisso de responsabilidade social e tornando possível estarmos aqui hoje. E um especial agradecimento ao BBVA por ter apoiado nesta sessão de encerramento. A HiTech é uma associação sem fins lucrativos que atualmente conta com 24 associados, cujo propósito é suprir uma falha de mercado resultante da escassez de iniciativas, com foco específico no apoio à translação de tecnologias desenvolvidas por investigadores para a economia e para a sociedade, seja através da criação de startups de base tecnológica ou de inovação aberta. Através do programa HiTech, pretendemos não só motivar equipas de investigação a testar a viabilidade comercial dos seus projetos de investigação científica, como capacitar os investigadores que participam no programa na translação do conhecimento gerado através do investimento público em investigação e desenvolvimento. O HiTech é um programa totalmente gratuito para as equipas de investigação que nele participam, o que releva o sentido de responsabilidade e compromisso dos 24 associados da HiTech que financia o programa. Este gesto de uma grande visão é demonstrador na clara aposta da HiTech em ajudar o nosso país na transição para uma economia baseada no conhecimento. A atual conjuntura faz-nos perceber cada vez mais a pertinência em apostar em ciência e tecnologia produzida em território nacional e, sobretudo, em transpô-la para o mercado. Apesar de sermos orgulhosamente uma iniciativa privada, temos uma missão com impacto público e acreditamos que juntos vamos conseguir. No entanto, a nossa intervenção não se circunscreve ao programa HiTech. Os nossos associados são convidados a apresentar desafios que necessitam de desenvolvimento tecnológico e a HiTech, recorrendo a uma extensa rede de investigadores capacitados no HiTech, procura apoiar a sua resolução. Estamos ainda a preparar o 2021 com um conjunto de novas atividades focadas não só no levantamento ou na oferta de conhecimento, como também na procura de conhecimento. O nosso objetivo passa por isso para atingir os 40 associados, que é o limite máximo de membros que podemos acomodar, mantendo a qualidade do nosso trabalho de proximidade com esses associados. Por isso, aproveito esta oportunidade para deixar o desafio a todas as empresas que acreditam no investimento em talento, em ciência e tecnologia ao serviço das empresas e das pessoas, que se juntem a nós neste grupo extraordinário. Termino desejando boa sorte às 7 equipas que apresentaram hoje os seus projetos, felicitando-os já pelo trabalho realizado e agradecendo a toda a vasta equipa que trabalhou com a HiTech para levar a cabo esta edição do programa e esta sessão de encerramento. Não deixo, contudo, de reforçar os 3 desafios. Aos que hoje terminam o HiTech, esperando pela vossa participação na fase seguinte, o HiTech Startup. Aos investigadores aqui presentes que têm ideias com base em ciência ou tecnologia, esperamos pela vossa participação no HiTech 2021. Às empresas presentes, juntem-se a nós nesta missão de aproximar ciência e tecnologia ao tecido empresarial. Muito obrigado, António Bradão de Vasconcelos.
Muito obrigado, Mário, por essas palavras do nosso presidente e pela sua disponibilidade para o substituir. Encerramos assim esta sessão e, como eu disse na minha apresentação, estamos a começar a preparar o ano que vem e o próximo HiTech que vai arrancar no primeiro trimestre de 2021. Muito obrigado pela participação de todos e até à próxima.